Hello, my name is Caden Matthews, grade 11. My project was the transference of a gene between two different types of bacteria. The materials used in this experiment were designed to create the petri dishes, connect multiple types of bacteria, encourage the breakdown of the plasmid and nucleus, and precise measurement tools that will allow for accurate results. The machine on the left is the vortex machine, used to mix the contents of a centrifuge tube. Meanwhile, on the right is a transformation machine, used to place a centrifuge tube at certain temperatures for periods of time. The first step of this experiment was to create the petri dishes that the bacteria would grow on. The first two petri dishes would not have Kanamycin, since it is an antibiotic that would prevent the growth of the bacteria on it. Therefore, when I inserted the gene into the petri dish that had the Kanamycin, it would grow because that bacteria was resistant to the antibiotic Kanamycin. This is what the gel looked like after being poured and allowed to cool for a period of time. First, I obtained a sample of E. coli DH5. I then trailed it on each of the two petri dishes that did not have the Kanamycin inside them. These would be used as a control and also a place to grow the bacteria elsewhere. I would then let them grow overnight. As the two petri dishes without Kanamycin grow, I would prepare a mixture that will allow for the genes to be transferred from the plasmid to the bacteria. The transformation buffer would work as a negation of sort that would disable the negative charges on the plasmid that would allow it to interact with the bacteria and enter it. The cold temperature, which will be the microcentrifuge tube will be placed into, will denature it when the sudden temperature changes to a hot temperature, which would open the protein tunnels. This hot temperature allows for the plasmids to enter the bacteria, which is a successful gene transfer, and then cooling it down closes those doors again. The LB broth would heal any damage that occurred when the sudden changes were breaking open the bacteria, which could cause lasting damages. And then when I place it at 37 degrees Celsius overnight, it would allow the bacteria to grow a bit and heal. The new bacteria mixture that was in the microcentrifuge tube would then be transferred onto the petri dish that had the Kanamycin. I would spread it out. That would allow for even growth all over the petri dish, and then I would allow it to grow at room temperature for the next week. On the left is my results on the 18th of March, where you can see the yellow dots are on top of the petri dish, which would normally be quite hard for bacteria to grow on since the antibiotic Kanamycin is there, which would usually negate antibacterial growth. And then a week later on the right is my results where some mold has grown, but that's normal because it's been left at room temperature over the course of 2 weeks. And this petri dish has really strong growth on it. And since it's growing over top of the bacteria, it's not being interfered with by the antibiotic. As I stated before on the last slide, the yellow dots show that there was successful growth and gene transfer from the plasmid to the bacteria. And again, I mentioned this as well, but the strain of BL21 is now resistant to the antibiotic due to the plasmid transfer, which allowed it to grow in the experiment. Now, this did not really prove anything, but it's similar to the process of the creation of insulin, where human DNA is inserted into bacteria, where the hormone is then created. It's similar because I inserted the plasmid into a bacteria, where it then allowed it to grow, even on a very difficult place for bacteria to grow on. This was somewhat difficult to work on, but due to the support of the molecular genetics lab at Seneca Valley, I was able to accomplish clear results.